what? Degradation of glycogen, gluconeogenic, synthesizing glucose, and ketogenic, synthesizing ketones. What are ketone bodies? What are these ketone bodies? Acetoacetate, no, acetone, 3 hydroxybutyrate. Okay. إذا هما mainly acetoacetate and 3 beta hydroxybutyrate. Okay, these are the ketone bodies: acetoacetate and 3 beta hydroxybutyrate. Okay, and proteolytic, يعني proteolysis or degradation of proteins to provide what? Amino acids for what? What do we need amino acids for when we make proteolytic? Source of energy. لأنه السينثيسيس هون أنا بعمل بروتيوليسيس، فمش معقول أعمل بروتيوليسيس and at the same time أعمل synthesis. Other proteins in other conditions. In the fed condition بيصير صح. بس هون proteolytic, since we are talking about fasting, proteolytic to use those amino acids for energy production. Okay? The strategy of metabolism is to store fuel when food is available and to mobilize these stores when necessary. Any question about these concepts? You must understand these concepts in order to understand the rest of integration metabolism. This is the insulin that, as I mentioned, hormones mainly control and regulate regulating metabolism. And here we are talking about insulin and its counter-regulatory hormones such as glucagon and insulin is a protein hormone. Okay, it is synthesized from or produced from beta cells of plant glands, and it is synthesized as pre-pro hormone. Then it will be processed into active. شو معنات pre-pro hormone? Yes. Okay, and it is inactive, and it is converted first to pro-hormone, then it is converted to hormone. Okay, after proteolytic cleavage of the C peptide, that connecting between two parts of the polypeptide chain. Once it is cleaved at this region here, insulin is left with 2 polypeptide chains, okay, attached by 2 sulfide bridges and 1 intrasulfide bridge. Okay. Where it is synthesized? In the pancreas. And where in the pancreas? And where in these beta cells?
nucleus وين يعني؟
Ribosome. Like any other protein. Like any other protein. Okay? The gene will be transcribed into messenger RNA, and that messenger RNA will be translated into cytoplasmic ribosome, okay, to give that pre-pro protein. Then it will enter endoplasmic reticulum and Golgi Golgi apparatus and to be processed to produce active insulin. Okay? يعني أنا I assume that you know these things عشان هيك ما بدخل في تفاصيل. Okay? Serial processing, okay, once it is secreted from Golgi apparatus. Okay? So when you have high glucose, insulin will be secreted, produced, as you see here. Glucose, amino acids, fatty acid will stimulate the secretion of insulin. Epinephrine will suppress its secretion. This is the action of insulin. Did you take these things before? Or is it the first time? Oh, you know some information about insulin.
About the regulation or the role of insulin in regulation.
And its structure and these things. Mechanism of action? So this is the mechanism of action. So I don't have to, if you know the mechanism of action. These are the counter-regulatory hormones, and you are talking about the glucagon and epinephrine here. Now remember that insulin is anabolic hormone. Glucagon is catabolic hormone. This is very important to remember. Anabolic hormone of insulin اللي هو بيعمل synthesis of glycogen، بيعمل synthesis of lipids، بيعمل synthesis of proteins. While glucagon is catabolic hormone، بيعمل degradation for glycogen، بيعمل lipolysis وبيعمل proteolysis.
ماشي حلقة النبيل.
الانسولين نفس الصورة اللي كده هو inhibited by epinephrine بس فهو لما يصير له inhibition بعدين بينقلب الجلوكاجون يعني الجلوكاجون ما بينقلب
ليش بيصير له inhibition؟ لما تركيزه بقل، اوكي، نتيجة الفاكتورز اللي عملت له سيكريشن انتهت وراحت، بيصير الابنفرين او الجلوكاجون.
بس هو ما بيعمل inhibition للانسولين الجلوكاجون.
الاوتكم ايه بيكون؟ انه when you have high glucagon or epinephrine you have low insulin. هذا الجلوكاجون okay and its mechanism of action via the G proteins and activation by cyclic AMP adenylate cyclase ATP to cyclic AMP. Cyclic AMP will activate protein kinase that will phosphorylate different enzymes, and phosphorylation of different enzymes will have different effects. Some enzymes are activated and some enzymes are inhibited. Who could give me an example on an enzyme that when phosphorylated is activated? Glycogen kinase. ما في
اسمه أطول من هيك اسمه بجد
phosphorylase kinase. Okay, وglycogen phosphorylase. glycogen phosphorylase على ايش مسؤول؟ breaking glycogen. When it is phosphorylated, it is activated. عكسه glycogen synthase enzyme. When it is phosphorylated, it is inactivated. Okay? And when it is dephosphorylated, it is active. So, as I mentioned, glucagon is a catabolic hormone that will break down carbohydrates, okay, and it will increase gluconeogenic, the body with glucose. It will break
إيش؟
يعني أنا ممكن أسألك في لما أتكلم هون عن الجلوكاجون it will break down glycogen, okay? So you have to know about the glycogen phosphorylase and how it is
اسمعي.
regulated. معلش هذا هو الانتجريشن.
لما أنا أحكي عن جلايكوجين
ايش ما في وقت؟
لازم تعرفي كيف الجلايكوجين بيعمل له ديجريديشن. لازم تعرفي كيف الجلايكوجين فوسفوريليز بيعمل ديجريديشن للجلايكوجين.
الجلايكوجين فوسفوريليز بيعمل
يعني ما اسالكم كيف بيشتغل
إنه
الجلوكاجون.
لما يفكه ويحط عليه فوسفيت.
هاي موجود هون. هذا بيشرح how glycogen phosphorylase is activated by glucagon.
إنه مثلا نعرف بس إنه الجلوكاجون بيكسر الجلايكوجين والإنسولين بيبني الجلايكوجين.
بس هيك بدك تعرفيه. خلص اذا جاوبتوا وظبطتوا حالكم معناته بتجاوبوا. أوكي. إذا حكينا هون يعني مثلا لما بنتكلم كيف it will inhibit fatty acid synthesis. Okay?
تحط ولا معلم.
glucagon.
بس أنا مش حاسة فهمت.
هذا واحد.
شو أنا بقول لك بعد المحاضرة.
وأيضا بيأثر على الأسيتيل كاربوكسيليز.
هي درست الفاتس رجعنا. خلاص يعني.
يعني هي والأسيتيل كاربوكسيليز. في fatty acids are released, okay, by the action of catecholamines and glucagon. Protein, there will be an increase in amino acid uptake by other tissues. يعني I will try to ask you. بعض المرات بننسى إنه تعرفوا how it increases or affects خليني أحكي gluconeogenesis أو glycogenolysis. يعني مش فقط تحفظوا إنه it will increase glycogenolysis, okay, أو الانسولين it will stimulate glycogenesis without knowing that it does.
خلاص اطلع من
خلص إذا أنتم جاوبتوا عليها وفهمتوها معناته سهل جدا إنكم تجاوبوا.
بس دكتور يعني إلا ما يكون اسم إنزيم معين كان بأي كان ولا شيء زي هيك يعني هاي تفاصيل.
يعني أنا بحب مثلا أجيب سؤال أوكي من 10 enzymes 10 enzymes وأسأل how they are regulated under specific metabolic condition.
طب كيف سهل يعني.
يعني بدك تكون عارف كل الميتابوليزم.
طب أجيب على الدكتور جيب على الدكتور.
على
إيش
كله.
إيش؟
كربوهيدرات وليبيدز وبروتين امينو
مين
اسيد.
كربوهيدرات؟
إحنا عنا. مين هذا؟ مين هذا؟ شكرية الكسب.
طيب خلاص
إيش؟
إذا
هذا
وصلتوا
صوته زي
لهالاشتراك،
كان صوته محنك.
I will consider. Okay. طيب لا بدنا نتكلم عن الميتابوليزم in different tissues under well-fed state. Okay. Well-fed state is sometimes called absorptive state. Once you take the food, digest them, absorb, okay? This is the well-fed state. It will take from 0 to 4 hours, digestion and absorption. Okay? What will be the result of this? If you have food intake, glucose, so insulin will be stimulated and insulin to glucagon ratio will be high. Okay? مهم جدا تذكر. Insulin to glucagon ratio is high,
شو الستوري
and
تاعت
as a
الجلد؟
result you will have hyperglycemia, hyperlipidemia, and hyperamino acidemia. acetylcholine. Okay, for the first 4 hours, from 0 to 4 hours. What will happen at that position? This is what happens in the liver under well-fed state. The first thing, glucose uptake in the tissues will be high in peripheral tissue, such as adipose tissue and muscle. In brain and liver, glucose uptake is insulin independent. Okay, there are receptors that will, receptors or proteins called GLUTs, GLUTs, okay? The glutes in muscle and adipose tissue are insulin dependent, and the glutes in brain and liver are muscle insulin dependent. Okay, so in liver, glucose uptake. Now we have high glucose, high amino acid, and high lipids in the blood. So we are talking here about these substances that will affect integration of metabolism, which are found in blood. So their uptake will be increased. Let's talk about carbohydrate metabolism under well-fed state in liver first. Okay, so glucose is uptake is high, and the first thing that will happen in the liver for glucose is what? Phosphorylation. This is the first step of what's happening for carbohydrate metabolism under well-fed state in liver. What is the enzyme responsible for that? Glucokinase. Not hexokinase. Very high concentration. Okay? So glucokinase with high Km. Okay, for glucose, it will phosphorylate. Now, insulin induces glucokinase. So, and the glucokinase is regulated by insulin. Okay? So, this is one effect of insulin in carbohydrate metabolism and well-fed state in liver. Okay? It will induce glucokinase. So, glucokinase will phosphorylate glucose into glucose 6-phosphate. Okay? What is the importance of phosphorylation of glucose? Trapping. Okay? Now this glucose 6-phosphate under well-fed state will go into glycogen. So glycogen synthesis is stimulated, okay, by the effect of insulin on glycogen synthase and on glycogen phosphorylase. Glycogen phosphorylase inhibited, glycogen synthase is induced by insulin as well as allosterically activated by glucose 6-phosphate. Okay? So this will go into glycogen, and some of this glucose 6-phosphate will go the hexose monophosphate shunt pathway. Okay? Why? Why? To produce an ADPH. Okay? And why we need an ADPH? For fatty acid synthesis. Okay? شايفين كيف coordination between these different metabolic pathways? Okay? So carbohydrate metabolism is coordinating, okay, for lipid metabolism. So some of the carbohydrates will go into hexose monophosphate shunt to produce an ADPH for lipid synthesis under the well-fed state. يس يس هي نفسها اللي هي الرايبوز ايش؟ بالدوز. Some of this glucose, okay, will also go into glycolysis, okay, to produce pyruvate. And why is that? Because the enzymes for glycolysis are activated by insulin. What are those enzymes? Fructose, PFK1, and pyruvate kinase. Okay, these are activated by insulin and by some allosteric mechanisms. So pyruvate will be produced. Pyruvate dehydrogenase also is activated allosterically and by induction by insulin. So pyruvate will be converted to acetyl-CoA. Okay? Now we have excess of acetyl-CoA, and they are precursors for fatty acid biosynthesis. So fatty acid biosynthesis will be produced by or synthesized in the liver by acetyl-CoA. What are the enzymes involved here? Fatty acid synthase and acetyl-CoA carboxylase. Okay. At the same time, fatty acid degradation is stopped by malonyl-CoA that is formed during the fatty acid synthesis. Okay? Now, a lot of fatty acid synthesis from acetyl-CoA that comes from carbohydrates as well as from the diet chylomicrons, okay? By lipoprotein lipase, they will be the triglyceride of chylomicron will be hydrolyzed into fatty acids and glycerol. Okay? But some of the acetyl-CoA will be used here Krebs cycle, ولكن very little. Okay? To have some energy for the synthesis of fatty acids, but not for production of energy, but mainly to see the storage of fuels, storage of carbohydrates as glycogen, and the storage of lipids as triglyceride. Okay? And this is what happened in the liver. Triglyceride will be formed from these fatty acids and some glycerol that comes from the glycolysis. Okay? So triglyceride will be reformed and packaged into very VLDL, very low density lipoproteins, and these will be transferred to adipose tissue. Why? For storage. This is coordination. This is an integration of metabolism. Okay? Amino acids. إذا تكلمنا عن carbohydrates, lipids, amino acids high amino acidemia. Okay? So الأبيض بيزيد كون من proteins يصير deamination for different types of amino acids. Some of them will be converted to pyruvate, or some of them to acetyl-CoA, and they will help or participate in synthesis of fats and formation of VLDL, as well as for protein synthesis. Why we need protein synthesis in the liver? Do we store proteins? No. So why do we need synthesis of proteins? Yeah, because we need some enzymes, transamination enzymes, glycolytic enzymes, PCA cycle, enzymes, fatty acid synthesis enzymes. All of these require some amino acids for their synthesis. Hormones of the liver, such as what? Yeah, I did remember also. If there are here we are talking about substrates, okay, that are used as a fuel for storage in case of well-fed state or degradation in case of fast. Yes.
دكتور انت قبل حكيت انه الفاتي اسيد ما بندخل كثير على سايكل كمية قليلة، هذا الحكي انه احنا بالليفر ولا بشكل عام هيك الفاتي اسيد؟
ليش ما بندخله فاتي اسيد على تي سي اس سايكل؟ الاسيد الكويل
الفاتي اسيد نفسه ما بنرجع نرجع او الاسيد الكويل
هو
معظمه بيروح بتحول لفاتي اسيد.
يعني هون الفاتي اسيد ما بصير بيتا اوكسيديشن للفاتي اسيدز لانه الانزيمز تبعت البيتا اوكسيديشن والكات 1 اللي بيعمل الترانسبورت they are affected by insulin. So no fatty acid oxidation.
قصدي لا الاسيد الكويل معظمها راحت على جهة الفاتي اسيد ما راحت على التي سي اي لانه احنا انسان ليفر يعني والمين خلينا نحكي فانكشن انه يعمل ستورج.
Yes.
يعني لو
يعمل
كنا في مصر
synthesis في المصل هلا بنشوف
في
Okay. Any other question here?
دكتور في معلومة حكيتها قبل انه الجلوت بتكون بالليفر بالبرين الانسولين اندبندنت يعني even if we are in the fasting state بضل كمان بنعمل trapping للجلوكوز هذا
يا بس بدخل الجلوكوز في البرين وبصير له ترابل. Yes. بصير له فوسفوريليشن.
بس احنا مش بنعتمد في الجلوت على الكونسنتريشن لما بدنا نوصل لهناك فاحنا اصلا عندنا لو concentration في الجلوكوز in the body.
بيدخل as much as the brain will accommodate of that glucose. هاي عن الانزيمز تبعت الجلايكوز.
بس الفركتوكاينيز وين هذا اللي كان بكرب السايكلز؟
مش فاهمين.
مش كرب سايكلز الجلوكوز.
وهذا ايضا بتكلم واي في الويلفيد ستيت ما في جلوكونيوجينيسيس بس اتليست لازم تتذكروا الانزيمز اوف الجلوكونيوجينيسيس. What are the major enzymes for gluconeogenesis? Pyruvate carboxylase, phosphoenol pyruvate carboxykinase, fructose 1,6-bisphosphatase, and glucose 6-phosphatase. الليفر فيه جلوكوز 6-phosphatase؟
الليفر فيه مصل
ليه؟
لانه مثلا ما بتكون نركز الجلوكوز
بس ما بيعمل
طب طبيعة الجلايكوجين
أو تستخدم
او
for energy production.
فكرة مزعجة.
Glycogenolysis حكينا عن phosphorylase والphosphatase دول لازم تتذكروا. And these are major ones. Okay? لازم تتذكروا في الجلايكوجين ميتابوليزم لازم تتذكروا الجلايكوجين فوسفوريليز والجلايكوجين سينثيز. جلوكونيوجينيسيس تتذكروا الاربع انزيمز. الجلايكوليسيس لازم تتذكروا PFK1 وPFK2 وجلوكوكاينيز وبايروفيت كاينيز. هذول لازم تتذكروا. في PFK2 لازم تتذكروا الفركتوز 2,6 bisphosphate which is very important modulator or regulator of glycolysis, increases glycolysis and it will inhibit gluconeogenesis. هاي عن الاسيتيل كويل كاربوكسيليز for fatty acid biosynthesis. Okay, how it is regulated by insulin and by citrate as an allosteric enzyme. ليش citrate? Shuttle, because it carries the acetyl CoA and transported outside and by citrate lyase تعطيه acetyl CoA وبيكمل for fatty acid biosynthesis. This is the adipose tissue in well-fed state. Okay? So glucose uptake is increased. Glucose will be phosphorylated. Okay? And pyruvate, and some of it in exhaust monophosphate chunk, or fatty acid biosynthesis for an ADPH, pyruvate by pyruvate dehydrogenase, okay? High incidence of pyruvate dehydrogenase is active, giving acetyl-CoA, some of it will go into PCA to produce some energy to help for synthesis. Acetyl-CoA is a precursor for fatty acid synthesis, activation of acetyl-CoA carboxylase and fatty acid synthase, okay, to produce triglyceride, some other chylomicrons, VLDL from liver, this is from diet, also by activation of lipoprotein lipases, okay, there will be degradation of the triglyceride inside here, then reformation of the triglyceride that using the glycerol that comes from liver, okay? To reform triglyceride, and this triglyceride will be stored when it is eaten. لأنه ما في جليسرول كاينيز بالأديبوز تيشو عشان نعمل ترايجليسرايد بدنا جليسرول فوسفيت، وبالتالي بيجي لك جليسرول.
اكتب important. There is no glycerol kinase in adipose tissue.
Amino acids uptake increase, protein biosynthesis, especially the branched chain. Amino acids such as alanine,
We take
leucine, isoleucine, and
we
valine.
take glycerol phosphate
دكتور ممكن بس تتكلم
From liver.
بالمايك؟
Excuse me, مش سامعين؟
لا
ولا أي شيء؟
ما سمعت ولا شيء بالنهار.
طب ليش ما حكيتي؟
رفعت يدي مرتين كنت قاعد تشرح.
لا لازم تحكي، لأنه أنا يعني ما شفتك رافعة ايدك، اوكي؟ وكنت اتوقع انه صوتي واصلكم كون العدد قليل او شيء. اوكي. So amino acid uptake for protein biosynthesis, glucose uptake is increased, okay? Glycolysis to make pyruvate, acetyl-CoA. Some of this glucose will go into glycogen for storage, okay? Now, some of the acetyl-CoA is used for oxidation and the production of ATP. Also, some fatty acids, okay, could be used in the resting case of muscle. They use, under normal conditions, fatty acids and some ketone bodies for energy production. This is what happened in a brain. Brain mainly under bifid state relies on glucose. تقريبا البرين requires about 100 grams of glucose per day, okay, for energy production. وهذا كله بيجي من الجلوكوز في الويل فيد ستيت، اوكي، دايركت، وبيدخل glycolysis as you see. Then acetyl pyruvate dehydrogenase to acetyl-CoA, it will be oxidized to produce high amounts of. Under normal conditions, ketone bodies are not used by brain, but under some stress conditions or abnormal metabolic conditions, ketone bodies will participate in feeding brain. Okay? Yeah. Fasting أو starvation اللي راح نشوفه. Yes. لانه ما بيصير لهم ميتابوليزم في الليفر على الترانس امينيزز تبعهم، لانه عشان نعمل ميتابوليزم بدنا اول شيء نعمل ترانس امينيشنز وشغلات من هذا النوع في الامينو اسيدز مش موجوده للبرانش في الليفر، فبروح على المصل.
اسألي جوزنا لأنه شوفي مش محطوط.
This is a summary
عند المصل
of what we mentioned.
محطوط بس عند الليفر والأديبوز تيشو.
هو بس يدخل الأديبوز تيشو تيشو فور ستورج والليفر
Um, um,
What organs?
combination of different organs, okay, with different metabolic pathways, liver, adipose tissue, brain, and muscle, and what happened in each case as I mentioned previously. Okay, so this is one picture that summarizes the integration of metabolism of different substrates, carbohydrates, amino acid, lipids, in different tissues as you see.
Okay. شكرا تسأل.
Okay, questions about the well-fed state?
What organs synthesize glycogen?
نتكلم عن الفاستنج state.
لأنه مش محطوط إلا الليفر والأديبوز تيشو برضه.
سنتسايز اللي هو إنه بيعملوا جلوكوز.
لا synthesize يعني بيعملوا storage.
These are concepts you have to remember before going to some details. In the absence of food, plasma level of glucose, amino acids, and triacylglycerol falls.
major cell.
Okay?
شكرا.
And that will take about how many hours? About 3-4 hours after the after the absorptive state. Okay? 3 to 4 hours. وبالتالي insulin secretion decrease and glucagon secretion will increase under these conditions. So low insulin to counter regulatory hormones, okay, and the ratio of insulin to those counter regulatory will be decreasing, and that will induce the catabolic metabolic state. What is the catabolic metabolic state? Degradation. Okay? Degradation of carbohydrates, degradation of lipids, degradation of proteins. Will stimulate glucagon secretion. Yeah. Yeah, yeah, yeah. Substrate are exchanged وهذا مهم جدا وهذا نوع من الانتجريشن. ايش هي substrate that will be changed? Glucose, fatty acids, ketone bodies, amino acids, okay, among liver, adipose tissue, skeletal muscle, and brain. Sustain energy metabolism in the brain, red blood cells, and other glucose requiring tissues. These are very important because these tissues, brain, red blood cells, and some other specialized tissues, they rely only on glucose under. فعشان هيك لازم هاي التيشوز coordinate with each other in order to maintain that glucose for these tissues. Pyruvate, alanine, and glycerol are needed for gluconeogenesis as non-carbohydrate precursors. Okay? البايروفيت شفتوا من وين بيجي؟ الالانين طبعا في هاي نعم under this condition alanine cycle will be active. Okay, يعني بيصير converging between pyruvate and alanine via transamination و والالانين بيصير to provide pyruvate also في هاي الالانين cycle in order to make glucose. Okay. Mobilization of fatty acids from triacylglycerol from the adipose tissue, white adipose tissue, for the synthesis and release of ketone bodies. عارفين البريكيرسر اوف كيتون بوديز ايش هو؟ الاسيتيل كوين that comes from fatty acid beta oxidation of fatty acids. Okay. Now, as you will see, under some starvation periods, the body will stop making a lot of glucose via gluconeogenesis because that will break most of the proteins of the body, and if that happens, that will destroy the body, that will cause death. So it will stop or reduce, not stop, it will reduce gluconeogenesis in order to produce a glucose. It will spare some glucose from gluconeogenesis to brain, and the brain will rely on ketone bodies, on beta hydroxybutyrate and on acetoacetate. Brain does not use fatty acids for energy production, but it could use, and it uses, the ketone bodies as well as glucose for energy production under long period of fasting or starvation.
هلا كنت حكيت انه احنا بنصير نستعمل كثير بروتينات بيسك علشان الانيميا، عشان يكون بيتا جلوكونيجي او قلل الجلوكونيوجينيسيس.
مش فقط عشان الألانين،
والاذا
كل other امينو اسيدز، كل الجلوكوجينيك امينو اسيدز هاي اذا استمرينا في الديجريديشن او البروتيوليسيس of the body proteins, will we end up with all our proteins, and if that happens, that is incompatible with life. Not only alanine, all glucogenic amino acids. Okay. هاي طفلنا عشان تشوفوا كويس. Okay, the liver maintains the blood glucose level during fasting state. Liver produces glucose via glycogenolysis. Why? Under fasting state صار عندي glycogenolysis. How it is regulated? By activating glycogen phosphorylase, because we have high glucagon to insulin ratio under this condition. عكس. Okay? فصار عندنا glycogenolysis. Okay? And this is what you see. Okay? For gluconeogenesis on the first 24 hours. Okay? Or even the first 10 hours, we have high degradation of glycogenolysis to produce glucose. So high concentration of glucose is coming from high rate of glycogenolysis, okay? And after 10 hours, it will be depleted. So by 10 hours, you are done with the stored glycogen in your liver, okay? So after that, gluconeogenesis from the alanine cycle, okay, pyruvate alanine cycle, or glucose alanine cycle, بصير عندنا gluconeogenesis after 3 days تقريبا بيكون the maximum، بس نبدا gradually. Everything is gradual exchange of substances between the different tissues. So liver produces glucose via glycogenolysis and gluconeogenesis for glucose requiring tissues. Okay? Gluconeogenesis. Why gluconeogenesis? لأنه الجلوكاجون activate gluconeogenesis, okay? And acetyl-CoA will not activate the pyruvate carboxylase. لأنه بيكون الأسيتيل كوي عندي قليل، وبالتالي بتبطل تعمل اكتيفيشن للبيروفيت كاربوكسيل اللي بتعمل اكتيفيشن، وبالتالي البيروفيت كاربوكسيليز will be activated. طبعا هون الميكانيزم اللي هو الجي بروتين والبروتين كاينيز A والسايكليك AMP by the effect of glucagon on protein kinase A and the glycogen phosphorylase. So in 10 hours تقريبا بنتهي، بيكون يعني الماكسيمم، وجراجولي in 24 hours بضلش ولا شيء من الجلايكوجين، وبالتالي لازم يكون في عندنا gluconeogenesis in the liver.
دكتور يعني الجلوكونيوجينيسيس بتبلش بعد 10 ساعات؟
لا، بتبلش بعد 10 ساعات، اوكي؟ وبتنتهي ب as you see here، وبيكون الماكسيمم in 3 days تقريبا، وبعدين بتنزل، وبعدين بيتكمل على بيزل الليفل. إذا استمر الستارفيشن بيكون عندي في gluconeogenesis، ولكن الريت of gluconeogenesis، وهذا measured by the amount of glucose produced، بيكون الريت أقل بكثير من هذا الريت. حكينا عن الفركتوز 1,6 بيسفوسفاتيز والإفكت أوف فركتوز 2,6 بيسفوسفات والبيبسي كي أند جلوكاجون. بعدين بصير increased fatty acid oxidation، بس ينتهي تقريبا الجلوكونيوجينيسيس مش معقول يضل مستمر. واي؟ لأنه بصير protein، so لازم يدخل another substance to help, and these are the fatty acids as you see here that will continue to some weeks. فالإنسان الobese could stay under starvation for longer period than the thin person. At the same time, ketone bodies will be produced to spare glucose, okay, from other tissues and for the brain.
بدون مي حتى.
الآن الجلوكونيوجينيسيس في under
بدون
these
مي 4
conditions
أيام.
على long term fasting
بس ما
أو
يرحم.
starvation بصير البرين حاجته إلى، يعني أصلا كان البرين بده about 100 grams of glucose per day، بصير بده glucose under these long term fasting or starvation 35 grams of glucose. The rest of energy production will become from hydroxybutyrate and acetoacetate and some amino acids that will go into gluconeogenesis for glucose. Yes.
Other tissues that is specific for glucose.
Like red blood cells. بنروح لها جلوكوز، اوكي؟ لانه في عندنا جلوكونيوجينيسيس ولكن مش بنفس الريت تبعت الايرلي ستارفيشن.
Low concentration of glucose
بيضلوا الجلوكوز رايح، ما هو هذا هو الانتجريشن اوف ميتابوليزم، انتو to sustain or to maintain energy sources for the required tissues. red blood cells will continue using glucose from gluconeogenesis, okay? Brain will reduce its rely on glucose، ولكن red blood cells cannot do that. some sparing of glucose to red blood cells. البرين بيوقف قليل من الجلوكوز، بصير ياخذ كيتون بوديز.
سبحان الله. أنا ما أهتم.
Increased fatty acid oxidation, and this is logical لأنه الكربوهيدرات ميتابوليزم تقريبا شبه ستوب، فبده يصير بيتا oxidation for these fatty acids as you see here. Okay.
شوفي ايش كان الوان.
خلصنا احنا؟
مثلا
طيب اعتقد نيكست لكتشر بنكمل عشان ابدا في الديابيتس والاوبيسيتي. يعني هو مش ضايل اشي كثير هون. هاي بتلخص كل القصه اللي حكيناها اوكي بالفاستنج ان ليفر، وبنشوف بالفاستنج ايضا شو بصير ان اذر تيشوز. اوكي بكون خلصنا الويل فيد فيرسس فاست سايكل والانتجريشن اوف ميتابوليزم، بعدين نبدا في الديابيتس اند اوبيسيتي. عشان تدرسوهم ولا ما تدرسوهم؟ راح يكون the 3 lectures about 10 questions. مع اللي بده يحضر اهلا وسهلا. اوكي لانه بالسيستم التدريسي اوكي هو not blended. I know يعني قدر بس this is the system. انت بتخلصي بكره باي ذا واي، يعني بكره انتوا مش عندكم محاضره؟ لما نكمل هاي واذا اخذنا شويه وقت بنخلص الاوبيزيتي والديابيتس. كلها عباره عن 24 hours 24 slides.
خلصها بكره اخذنا
عادي لو اخذت ساعه ونص.
طيب يلا يعطيكم العافيه.